Going to get back to it. All right, I'm going to go quickly now because we got all the big questions out of the way. This is now just the details. So we got the directionality, suggesting some kind of time sequence encoding. This is just a little video showing. This is sort of what O'Keefe would have seen. And this is the raw data. You're basically seeing cells. You can see this is spikes as they're being emitted as the animal's moving. So this is not something that, a representation that comes out of massaging, the data. This is the raw data. And what's interesting is now as the animal's moving, when the animal stops, if you just listen, the animal's gonna stop, a little pause, and then you get this little burst of activity. Burst. Now when the animal moves, if you listen, you're gonna hear this rhythmic modulation, the theta rhythm. It's this kind of Now we'll see how. See, my ears are sort of trained so that I can just pick this out. It's like right there. Can you hear that? It's this rhythmic modulation as the theta rhythm, and that rhythm goes away when the animal stops. So you really have these two modes, the theta and the non-theta mode. Theta associated with moving, non-theta associated with stopping. And this is just showing, this is the raw spiking activity. If you take another approach, you just use kind of a simple Bayesian decoder, and that is, if I know the firing rate is a function of position, I can then invert that, compute conditional probability that the animal's in a given location given the firing. So I just every 200 milliseconds we just see which cells are firing, and then we ask the question, what's the probability that the animal's at any location on this track, using Bayes theory, what's the probability that's at that location on the track given this pattern of cell firing? And the probability is going to be shown as the triangle. Magnitude of probability is the size of the triangle. And so this is the application of a simple Bayesian decoder, same data. This is the green circle, that's where the animal actually is. And so what you see is big triangles where the animal is. There's no triangle. Now there are actually triangles everywhere. Every 200 milliseconds, you compute the probability at every location. It's just the triangles at every other location are essentially zero. The hippocampal, representation really reflects the animal's current location, except the animal stopped. Triangles go away, and now during those bursts, you see these triangles jump around on the track. When we zoom in on this, what we'll see is that those brief periods of immobility correspond to a transition from the hippocampal representation, or code, if you will allow, to represent, rather than represent the animal's current location, represent remote locations, and in particular represent trajectories of remote locations. We'll see that the animal is replaying sequences of locations at remote locations on the track. Yes? So people have looked at that to ask whether the content of replay is in some way correlated with the direction of motion. In part, it was as I'll kind of get to, there was this discovery by Dave Foster when he was in my lab of this so-called reverse replay that occurs. Animal walks to the end of a maze, and when he stops, you get, reverse replay. But the animal could also turn around and pointing in the other direction, in which case you can get forward replay. So if the animal, like at the starting point, is pointing this direction, you get forward replay. Animal's pointing this way, you get reverse replay. And so the question is like the forward or reverse replay tied to, the heading direction. And the answer is no, that it's not really tied. That's you can get both. Could it be informed by, by sensory information coming in at the time? That's interesting because, as I'll point out, these replay events that you see during quiet wakefulness look very much like replay events during sleep. And replay events during sleep can indeed be biased by sensory input. This is this auditory cueing, so pairing auditory cues by delivering sensory information to the sleeping brain that can bias the replay events. And so the question is a similar bias available and applied during the awake replay events? Very probably yes. It's just that that bias is going to come in the form of whatever the entorhinal, sort of the biased entorhinal code. And so it's just not as simple as the head direction. But, again, it's like the directionality, the local view thing. It's like directionality of place cells is not sort of simply explained by direction of view. It really is going to be like the product of the path integrator. And so that will be like the simple model that I actually present that Foster originally, proposed. It was that. It's the path integrator. Whatever the state of the path integrator is, that serves as the initial condition for the expression of the replay. And so there was a very clever experiment. This was one done by someone that we all know and love because they are sort of tied to CBMM, but also embrace the sort of the computational biological framework, and that's Demis Hassabis, who founder of DeepMind, just like real success story. But he actually was a behavioral electrophysiologist as well, did experiments recording in rats, and did an experiment on observational learning. And so in this case, the question was, can you bias replay based on what animals are looking at? Here's a rat is watching another rat, and what he argued and what he demonstrated and argued was that the replay of rat 1 was biased by rat 2 getting reward location. Now, one interpretation of that is that, it's biased, but it's biased based on what the animal is seeing, right? It's not like rat 1 is imagining being rat 2. It's just, right? It's the sensory information that is getting, as a result of observing rat 2. And so that would be like consistent with what you're arguing. That is, there's a bias that on replay. Now you can ask, what is actually, when you're, in that observational experiment, what is actually being, what is the bias? Is it the heading direction, or is there like a path integrator version of observational learning? And that is, you're watching the transition in states. It could be like the change in, it could be a head directional self-motion like signal, or it could also be you're kind of integrating the state of an observed individual, and it's that serves as the bias. So it's like the remote path integrator model, not just the head direction, it's not just the location animal, but it's like integrating what the animal did. And this would kind of fit with, ideas about certain parts of the cortex that can, these mirror cells and other things that can, reflect, self-motion as well as equivalent motion of a remote agent. It would be the same sort of idea. And I think that's probably reasonable. So the short answer is, I think no on the head direction, but I think yes on the kind of the actual observed state probably does bias that. Oh yeah, and then there's like the sleep. There's sort of the sleep. This is just a movie, and you've seen it. So it's the same thing. Do the decoding analysis, but now the animal's taken out. It's not in the maze anymore. It's sitting in a little box. You can see in the inset, and this was on loop. But you can see that you get the same kind of replay-like events on the maze. You can sort of decode sequential states on the maze even though the animal's not there. So this is the so-called sleep reactivation. And so this is now the kind of the hippocampus online and offline. As I referred to there, you can think of the hippocampus being these two states. There's the active, taking in of information during interaction with the world, reflected in the sort of the macroscopic brain state here, in the form of these oscillations. So you get these oscillations around 10 hertz, the so-called theta oscillations. Actually, the theta oscillations, you can kind of refine that. Thinking about in terms of frequencies can be a little problematic because the theta and the range can go from like 4 to 12 hertz. In humans, theta oscillation tends to be a little bit lower, closer to 6 hertz. When we talk about it in rodents, we talk about it closer to like 8 to 10 hertz. But there's also this, an observation that theta itself, not monolithic. There's sort of a classical reference to so-called type 1 and type 2 theta. Type 2 theta being a sensory evoked like theta. Type 1 theta being one that's expressed during active locomotion. They are driven by different sources. They have different sensitivities to neuromodulators, and whatnot. And the locomotive theta is a higher frequency theta. That's the sort of the 10 hertz theta. The sensory theta is lower frequency. So you can kind of think of the dominant, what the dominant frequency of theta is expressed could depend on, which of those, the contribution of these two sort of theta oscillations. But the idea is that there is coordinated activity at a time scale of, on the order of, 100 milliseconds or so, that carries out some critical computational function during active interaction with the world. It could be sensory attention, it could be active locomotion. When you're moving, clearly you better be paying attention. You should be paying attention because, stuff's happening and you need to be able to react. So the locomotive condition, you could say, is, while it might not be necessary for theta, it certainly is sufficient. That is, the world's changing, and you should be sampling at high rate. Whatever you're doing, sampling at high rate. Sensory theta, okay, you should be paying attention. Maybe you can do it at a lower rate. Again, not necessary, but may be sufficient for like this sort of passive processing of attentional processing of the world. And when animals enter into the state of immobility, as you mentioned, where immobility is like, okay, they just stop moving. But stopping moving, in this case, you can see there's like a half-second transition in the so-called sharp wave ripple state, which was classically referred to as the LIA state, large irregular activity. And that's because you see these kind of irregular periods of sort of large deflections in the local field potentials, which suggest like there's massive input, synchronized input into the cells, because these field potentials largely reflect the result of coordinated input. It's not really spiking output. You sort of think, oh, look, lots of cells must be firing here to get these big deflections. Well, there's actually lots of cells are getting synaptic input to get these deflections, and there might be spiking that goes on there. And so you get this clear transition, but it's very possible for an animal to stop and enter into an elevated theta state. This is so-called vigilant state. So if you can be immobile, but totally freaked out, waiting to get eaten, and what you don't get this LIA, right? You get heightened theta state. Similarly, even though it's sort of infrequent, and an animal just before it stops, if we really like put the actual, velocity here, just before stopping, there can be like a lead or a lag in the expression of theta. And so you can kind of, you can make the transition into the non-theta state slightly before you stop. And similarly, you can enter into the theta state slightly before you begin moving. And so it's not really, the movement is a reflection and can be used as a proxy, but it really is, you can kind of think of it as like the attentional state. When am I paying attention? When am I not paying attention? If I'm not paying attention to the outside world, that's non-theta. If I am paying attention, that's theta. And that can be when movement is sufficient, but not necessary. And so I'm going to focus on this theta state. And so the idea that, like, why would you even need these, why would you need these oscillations at all? What function do brain oscillations serve? And thinking about like synthetic networks, they don't have any dynamics. They have no oscillations. I even raised this with Dennis when he comes to visit, and he was very skeptical of the idea of like sort of temporal coding or any kind of fundamental necessity for like temporal processing. And my argument was that like these brain dynamics, by their very nature, sort of introduce and impose like a timing constraint, sort of an analog computational temporal constraint. And so the question is this important? Is this used? And, what is the value of imposing a timing constraint? And that's what the dynamics are. It's at certain times, there's like more or less input. Lots of cells are receiving input here at the peak of these oscillations, because that's what the oscillations reflect. They reflect a period of like more excitatory input. These reflect periods of more inhibitory input. So you're kind of controlling when cells are firing. And so what would be the value of that? Well, kind of go back to an observation John O'Keefe made, as I mentioned. This was in early 1990s, 1991, where if rather than just look at the location the animal is at, like how many spikes are fired based on the location of the animal? And this is where you get place cells. But he asked, when the spikes start to fire as an animal enters a place field, what's the timing of it? When are the spikes, expressed relative to this underlying theta oscillation? That's the phase. So if we take the peak phase of this theta oscillation here, shown in red, and then look at the timing of spikes in red, this is the hippocampus. This is like in the prefrontal cortex. I'll mention this later. Just ignore that for now. And so what O'Keefe observed was when the animal first enters the field, the first spikes are expressed here late, right at the peak. The further into the field, now this is like time, but it's also space if the animal's like moving. The spikes occur, you get more spikes, this is the increase in firing rate, but there's also a shift in the phase. The spikes are emitted earlier and earlier. So as the animal enters the field, what you get is you're getting this kind of pop, 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 right? More firing rate increases as the animal enters the field, but also the spikes are firing earlier and earlier in phase. So you get this pop, 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 pop. This so-called phase precession. This is what John referred to this as, phase precession, as the shift in phase as a function of change in distance into the field. And you can illustrate that here. This is actual data. And so this is the position. So the animal's entering into the place field. So we just look at the overall marginal distribution of firing as a function of position. This would be the place field. Lots of spikes here, not many spikes here. And so the firing rate would look like this. But this is now the phase. Each and every spike is a function of the phase of the theta oscillation. So what you see here is the animal first enters the place field. The spikes are expressed here late in phase. And as the animal goes into the place field, they're earlier and earlier. You're getting more and more of them. A couple of things are happening. More spikes, earlier phase, but also more variability. It's like spread out all over the place here. And so this is the phase precession, profile of a particular cell, very characteristic. Question. So the oscillations that you see, like you put in the local field potentials, are the result of the kind of the synchronized, currents generated by population of the cells resulting from synaptic inputs. So the theta oscillation as measured by, local field potential electrodes is really a reflection of time-varying inputs that the cell is getting. Lots of cells. So it's just like it's an average measure of what's the local population of cells, what's the timing of the inputs that these cells are getting. These are all place cells. So the cells that are theta modulated, place cells are going to be theta modulated. So that's why, you look at the individual place cells, when you were listening to that, like when you're listening to the video and you could kind of hear the ch-ch-ch-ch, every one of the, the cells participating in that, those are all place cells. And it's because they were all being theta modulated that you could hear in the population this rhythm. Well, because there's two things there. One, so you could say the most obvious, why are these place cells firing in a theta rhythmic fashion? And it's, well, the inputs are modulated by the theta rhythm, right? The inputs are rhythmic, so obviously the outputs are going to be rhythmic. But then you can ask the question, why are the inputs rhythmic? What's generating the rhythm? Is it individual cells? Is it the result of a pacemaker circuit? So there's been a lot of work looking at, the generation of the theta rhythm. And, the conclusion is that they both are intrinsic properties. Individual cells have currents and conductances that are capable of, resonating or even expressing a theta rhythmic-like modulation. But the population expression of the theta rhythm really requires input from multiple sources. One, you need real input, like from the entorhinal cortex, for instance, because you're not going to see these. You're not going to see the currents unless you have inputs. But also, there is a modulation of excitability that comes from, in particular, the cholinergic nuclei in the basal forebrain, and the medial septum is a primary source of cholinergic input. So acetylcholine, elevated acetylcholine levels are associated with the expression of the theta oscillation. If you remove or lower acetylcholine levels, you get a transition into the non-theta state. But this cholinergic projection from the medial septum is accompanied by an inhibitory, a GABAergic projection, and the GABAergic projection is also oscillating. So you can think of that this oscillation is both, oscillatory excitatory input, neuromodulatory input that is like giving the circuit resonance to saying, look, you should be receptive to theta oscillations, and oscillatory inhibition, which is kind of structuring the timing of both the integration of the excitatory input and the timing of the spiking output. And so that's sort of what this model is intended to reflect. And that is, this is a cartoon model in which the red indicates time-varying inhibition. Red is the time-varying inhibition. And in blue is this like path-integrated spatial input. Now, this is just kind of a simplified version. The actual version would include, so the blue input is also theta modulated. It's also time-varying. And so if you were to include that, there's certain interesting things that come out of that. But we'll just go with like the simple model. It says the excitation is varying as a function of space. Inhibition is varying as a function of time. And if you combine those two things, and then you apply a very simple biophysical rule, which says, when is a spike going to be emitted by a cell? It's going to be when excitation exceeds inhibition. That's all. And so when does that happen? Well, when excitation is low, you have to wait until inhibition gets all the way down here before you can produce a spike. So, not a very complicated principle. If you have a weak input, it takes longer before you get a spike. The stronger the input, earlier the firing. And so that's what you get. As the excitation increases, the blue curve, exceeds the red curve earlier and earlier. So this is sufficient to actually give you the phase precession, the properties of phase precession to see here. Both the sort of the linear-like correlation of phase and position, but also if the idea is whenever the blue curve is above the red curve, whenever excitation exceeds inhibition, you can get spikes. Well, you notice what's the time period here when you're, weak input means very little time. There's very little time that blue is above red. As opposed to here, high excitation, blue is above red a lot of the time. And that means it can fire any time in this interval, which means here at these early phases, when it's firing earlier, it's also going to be more variable. You're going to get more spikes and they're going to be spread out over time, which is exactly what you get here. And one way I like to think about this is actually, if you break this down, you think of this, phase precession is like this linear progression. Well, I don't know. If you just forget about, let's just split this in half. So we're going to look at this half, right? This could be either in phase or in position. So the early part of the place field, this looks pretty linear. It actually is, pretty good. There's a pretty good correlation of phase and space. If you look at this half, though, if I block this out, and I just look at this, sorry, but that doesn't look it's not very well correlated. Phase is all over the place and gives you very little information about relative position. And so what you seem to have, but what you get here, lots of spikes, very little temporal information. Here, lots of temporal information, very few spikes. I like to think of this as the temporal coding, domain. This is the temporal coding region of the theta, of this sort of theta oscillation. And this is the rate coding region. And so here, when you're getting lots of spikes, well, I can't tell you relatively where you are here. I can tell you that you are actually in this place, right? I can use the number of spikes to express or reflect, for instance, sort of degree of confidence in the spatial estimate, the likelihood that I actually am in this place field. Or I could use this for, as we were discussing before, like the rate remapping, rate modulation. So I can use variation in firing rate here, and it's not going to be conflated with this temporal coding. Here, few spikes, but because there are very few spikes, it means I can position them precisely in time. So I can use the low firing rate properties to give me more precise temporal information, and the high firing rate properties to give me more, sort of rate variability or rate information. And so this applies both in space, so the early part of the field, temporal, late part of the field, rate, but also in phase. And so if I cut this in half this way, the late phase of the theta oscillation gives me good temporal information. The early phase, if I just look at this part, it's like I'm getting no temporal information, I'm getting all rate. And so it suggests that you kind of split the theta, this sort of theta oscillation has sort of two phase coding regimes, rate, temporal coding. And so that says that phase, relative phase of theta can matter. Now we can show, when I get Josh Siegel, a graduate student in my lab, using optogenetic manipulations, demonstrated that indeed, if you selectively manipulate hippocampal activity at these different phases, the early or late phases, you can demonstrate that they have different behavioral consequences. They're sort of different, they serve different functions. But first, so this observation, again, John O'Keefe made this observation early 1991. I termed it phase precession. But what's phase, what does it buy you? Okay, it's interesting. You say, oh, look, a temporal coding. What does temporal coding really buy you? And I think it really becomes clear when you think about how phase precession transforms hippocampal representation, not for single cells, but for multiple cells. So here, the example is 2 place cells. Each have phase precession. And now you can ask, using the simple model, like when does the place cell 1 fires, here, place cell 2 fires just to the right of it. And so there's like a spatial order, place cell 1 followed by place cell 2 as the animal runs through here. First place cell 1, then place cell 2. But if you ask when are the spikes for each of these place cells emitted, because of this kind of gradient-like property, the ramping property, this is the sort of the x dot equation of motion, path integration-like property. The spikes, the red curve is always going to be above the purple curve, which means spikes from place cell 1 will always fire before place cell 2, 1, 2, and it will do it each and every theta cycle. So every theta cycle, you're getting 1, 2, 1, 2, 1, 2, 1, 2. That is a recapitulation of the spatial order in the temporal ordering of spikes. And it has 2 interesting properties. One is it eliminates absolute time. Like how much time does it take the animal to go from here to here? I don't know. Depends on how the animal's feeling. Could take 1 second, could take 5 seconds, could take 10 minutes. Variable behavioral time scale is variable and unpredictable, right? Now, it doesn't change the relationship here. Place cell 1, place cell 2. This position is always going to be to the left of this position. The time it takes you to actually, sort of traverse these can be variable. What this does is it eliminates that. It doesn't matter if you take 1 second, 5 seconds, 20 seconds. You're always going to get this ordering. And so it removes the variability of behavioral time scale, and it puts it into a fixed biophysical time scale. So this the spatial order is now transformed into this 100 millisecond time scale. So this, you could say, why the theta oscillation? Why 100 milliseconds? So the time scale of the theta oscillation imposes a timing relationship between these sequential states. So this cell was going to fire, like 5 milliseconds before this cell. Why is a 5 millisecond, on the time scale of millisecond, 10 milliseconds, why is that order going to matter? And sort of Mayank Mehta, when he sort of made these observations, formulated this model, posited that, well, this kind of fits with the notions of spike timing dependent plasticity, that both the time scale and the asymmetry, and that is that you can strengthen the connections between 1 and 2 if the order is like one before 2, and that the time between those is on the time scale of about a few milliseconds. And so there's a very precise spike timing dependent plasticity, has very precise order and timing constraints. Phase precession seems to impose the constraints that would be sufficient to drive a spike time dependent plasticity mechanism. So this would be a good way of capturing or encoding using synaptic plasticity exactly these sorts of order relationships. Now, that doesn't mean you're going to connect place cell 1 and place cell 2, because this is in CA1, and in CA1, the number of connections between place cell 1 and place cell 2 is essentially 0. They're not directly connected. But downstream, the convergent target, whoever's listening to place cell 1 and place cell 2, are going to receive inputs 1, 2, 1, 2, 1, 2. They're going to get that order information. Yeah, question? Well, there are a number of great questions in there. So one is like, what happens when, so here where there's like sort of a single predictive trajectory, right? Okay, that makes sense. And in a sense, this is used, the linear track was used to constrain those trajectories, not because that was necessary, but because it allowed you to most sort of clearly demonstrate the phenomenon. So what happens when you have a branching track? And so an animal comes up to it, now you have 2 paths. Well, what happens? Well, one interesting thing, again, this is sort of the challenge to the directionality and local view hypothesis. What happens when you actually get to a choice point is the place cells will now become sensitive not only to location, the fire at the choice point, direction, like which way am I facing, but also where am I going to go next? And they become trajectory dependent. And so there'll be a place cell that will fire when the animal is going to go right, and a different place cell will fire when the animal is going to go left. So you actually get, it's like, again, this sort of contextual, code. It's like, everything exactly the same except where am I going to go next? And that's enough to remap place cell firing at that choice point. So what happens to the, this whole phase precession model? What you get is on different theta cycles, this might be place cell, place cell 1, like center, and this might be so maybe this is like the center arm, this is the right arm. The left arm is going to have another cell, place cell 3. And so what you'll get as the animal approaches this, you'll get some that will go one, left, one right, and you can get this on essentially adjacent theta cycles. You can get a flipping back and forth between the two. Now there's some, I don't know if I call it controversy, but there's sort of contention of, regarding the relationship between the bias in the, the theta, this kind of theta sequence and actual choice behavior. Some argue that, oh, look, you see the theta sequences that correlate with the animal's future behavior, therefore, which it does. But in part, that's because the place cells that participate in the theta sequences are biased by the animal's future behavior. So, it's unclear, does the theta sequence reflect what the place code already expresses, which is, likely the case, or is it the case that, if you could get the place code to actually flip, or the theta sequence code to kind of flip one direction, could you get the animal to, show that the behavior goes with the theta sequences rather than with the place cell bias? That hasn't been demonstrated. Now, let's see, there was a. Oh yeah, at the first trial. So Dave Foster actually asked this question. So when you see these, now there are two things, there are two phenomena that are shown here, and I sort of put them together. One is the phase precession. Phase precession is a single cell phenomenon. What I'm showing here is like phase precession coordinated across a population. And if you extrapolate this from 2 cells to a whole population, what this says is that phase precession, when you combine it in a population, will give you a sequence, what we refer to as theta sequences. Lots of cells, place cell 1 to 10,000, all firing in a sequence. And so what you get is, and if you decode this, as we'll see, what you get is like a trajectory. The population is following this. And what David Foster pointed out is that phase precession emerges right away. Theta sequences require experience. And so it's the idea that, phase precession might be a property, sort of derived from the intrinsic biophysics of the cells, but to get the population to actually all precess in the right way, because there's no guarantee, just because place cell 1 is fired, that place cell 2 is fired. Now, one way of thinking about that, it's back to this, the temporal coding. The phase precession, the sequential property, is largely a consequence of this temporal coding, right, relationship. But that's in the low firing rate regime, which means very few spikes. So it means that on any given theta cycle, so theta's, sort of the theta cycle, theta's maybe 10 hertz. So you're getting, if a cell that we're firing in, every single theta cycle would be firing at about 10 hertz. That's what you get. But that's like sort of in the peak firing rate. That's a cell firing every cycle. Most cells don't fire every cycle. And so in order to get a robust theta sequence, you basically have to, like in this theta cycle, we're all going to fire, right? We got our low probability spiking. We're all going to fire low probability in this theta cycle. And so there needs to be some coordination to get participation of enough cells to give you high fidelity sequence encoding. And that seems to require experience. And so it kind of makes sense. It just says there's like a plasticity mechanism that increases the coordination of the population. Yeah, that's the thing. So you're getting rate encoding information here. So here at this point, the high firing rate, you're getting basically at 10 spikes, you're getting a spike every theta cycle. So here, this like the early phase portion, getting, all the cells are firing at this early phase. So in early phase, you're getting lots of cells firing. That's the firing rate regime. Exactly, so that's the question. So you say, look, if I just had if all I want to know is firing, where am I, right? I just need firing rate. In fact, firing rate's going to be more useful because I have lots of spikes. I can like give you a confidence measure. It's like, look, I got like 50% of the, the inputs are like half as strong as I would expect. I'm producing 50% of the spikes, so I'm 50% confident. So you could actually grade the relative firing rate measure if what you were trying to do was just answer the question, where am I? Or am I where I am? So you could say that's like a recognition question. Is this something I recognize? Is this? Now you could ask another question, just thinking about, sort of standard memory measures. The simplest, the simplest memory query is a recognition memory. I show you a picture, you remember this? Like, oh yeah, okay, I'm 50% confident I've seen that before. The harder one is like a recall. Okay, I show you this, what were you doing before this? Or what were the pictures that followed from this? So now the recall requires that you actually, again, kind of follow this sequential chain. And so recalling temporal associations, which presumably would come from, this temporal coding things, connecting 1 to 2 rather than just saying, oh yeah, I know where I am. And so you can think of this as like recognition, recall. In recognition, you can think of this is like the contextual thing. Am I in lecture 1? Am I in lecture 2? Is this the morning? Is it the evening? And so kind of establishing the map, identifying the context, could be a product of this. Once you're in the context, it's like, okay, now what's happening? What's changing? What's the episodic content? How am I interacting with that context? That would be here. And so you could take this, and so I think what's interesting about this, you can say, well, look, if you can really divide it up in that way, by phase, late phase, episodic, early phase, context recognition, then you should be able to do manipulations of activity as a function of phase and show that different phases have different functions. And that's exactly what Josh Siegel, when he was in the lab, did using closed-loop genetics. Just block activity at different phases and ask, what's the consequence of that? But if you take this idea, so you got like phase precession, single cells, you've got the theta sequencing, which says, okay, coordination across cells. And now if you take this back to the population decoding level, and so you do the same thing that we did with like the triangles, Bayesian decoding, but now Bayesian decoding not at 200 milliseconds, but at 20 milliseconds, so very high resolution decoding. And so now you see the real, or at least a more real reflection of the sort of the hippocampal spatial representation. And that is, now you see, this is the dotted line is the animal's actual motion. This is like the decoded positional estimate. And so you see the decoded positional estimate actually sweeps. It's not at a fixed, it doesn't track this. It actually kind of follows this, it kind of sweeps from just behind the animal to just in front of the animal with a characteristic slope. And if you average all these things together, you take every theta cycle, what's the average, positional estimate relative to the animal's present head location, what you see is that this is the hippocampal code, if there is one, and that is every 100 milliseconds or so, it kind of goes from just behind the animal to just in front of the animal. It kind of sweeps in front of that with this sort of peak right here near the present position, which is sort of like the, it's like the rate code. That's the place code. And so 10 times a second, you're actually kind of sweeping from behind and front, past, future. And it's tied to the phase of the theta cycle. So this kind of late phase, you see the late phase in the theta cycle associated with like future position, the earlier phase associated with present location, which is exactly, that was sort of the idea here. Late phase, temporal code, it's like where am I going? Early phase, rate code, where am I? And maybe like, where did I just come from? So if we take this idea, that's the hippocampal, so the hippocampus, when it's interacting with the environment, put this rhythm in, now you're capturing, with experience, you're kind of capturing these kind of local temporal sequential relationships. I'm capturing, where did I come from, where am I going to? And so you're building somehow maybe using like these spike time dependent plasticity mechanisms, a representation that can do this predictive coding based on where I am. And so if you start from that, you say, oh, okay, so the hippocampus is coding experience in these short sequences. And you can take the time scale, so this is like 20, in this case, like 20 or 30 centimeters. It's about like 1 to 2 lengths of an animal, that's of the rat. How long does it take to go through that? Again, behavioral time scale is variable, but we're talking on the time scale of seconds. And so, the hippocampus is essentially capturing experience in these short second or two little chunks. I've referred to this as the sort of the MTV model of memory, but nobody watches MTV anymore. So, MTV was like these music videos, and the thing about music videos is like nothing long, right? It's just short, like quick transitions, short little snippets. So everything is edited together, and that's kind of the the way you experience an MTV video. And in a sense, that's sort of the way in which rats are experiencing the world. It's like in these little short snippets. Now, you could say, okay, that question. So there is a correlation. So theta frequency does increase with running speed. It's not a super high correlation. Actually, it's sort of theta power is actually more correlated with with velocity. But there definitely is there's variability in theta frequency. I don't know if I'd make too much of that. I think that there are sort of interesting observations that you can make regarding this kind of coding mechanism, and that is that essentially the sort of the length of in kind of time, how far forward you project, is a function of the size of the place fields. If you have small place fields, this is basically, so I said like 2 animal lengths, it's really more like sort of 2 place field lengths. So whatever the size of the place fields, that's how long the theta sequence goes. Big place fields, the sequence will span a lot of time. Small place fields, less time. And that's interesting because what you tend to find is like place field size seems to correlate with things like, reward, behavioral variability, where there's lots of stuff going on. Anywhere there's a lot, you have to do a lot of things, that the place fields get smaller, which means the sequences are going to get smaller. Where you have large stretches, things that are less interesting, the fields get larger. And so that's where you would, you could say that's like the resolution. It's like, do I really need to sort of do, prediction, every 20 centimeters while I'm walking down a hall where nothing interesting is going to happen, for 20 meters. And so that's where the theta sequences could actually scale. And so that's where things like there's been some, work looking in bats to see, bats, during real, sort of navigation where they're spanning distances of, not centimeters or meters, but kilometers. And what you find is the place fields kind of expand. What that means, the theta sequences would also now be, they wouldn't be 20 centimeters long, they would be, like a kilometer long in principle. So it's kind of an interesting way of thinking about how the temporal sequential representation really captures the spatial scale, and the spatial scale capturing information about kind of likely contextual variability. Is something going to happen? It's kind of back to like the remapping question we talked about. You get remapping or change in place fields when something, let's say when predictions are violated, when expectations are not met, when you feel the need to introduce a new representation to capture new contingencies. And so that's sort of the idea, the theta sequence giving you, it's like a predictive time scale. When is, how long can you go before the unexpected happens? That's the way I really think about it. And so that there's kind of a fundamental time scale. You can say, and the hippocampus is saying here, on the 20 centimeter time scale, the time scale of like 1 or 2 body lengths, I'm like 99% certain what the next state is going to be. Further out than that, not so sure. And so that can be, variable. And I think it's interesting thinking about like computational models where you can embed that. So you embed like a representation, a temporal representation that captures this sort of predictive uncertainty. And that is that there, you can kind of embed in the model representations like this when there is that certainty. And so you don't have to waste your time like doing unnecessary prediction if, And you can do that in this case by essentially scaling the receptive fields of individual place cells. Okay, and then I just quickly show you say, okay, so you get these representations. Back to this notion of coding, which I've tried to be a little, careful in using, because a code requires a decoder. Somebody downstream's actually got to pay attention to this, or otherwise it's not a code. It could be a representation. And so, thinking about like this theta representation, who's downstream of the hippocampus, and what would it, how might it be used? Sort of a key downstream, partner of the hippocampus is the prefrontal cortex. Actually, both hippocampus and prefrontal cortex lie at the top of a processing hierarchy. If you kind of go from like low-level sensory areas, you go forward, and low-level visual cortex goes to higher-level visual cortex, goes to like polymodal association cortex, and then ends up going either prefrontal cortex or hippocampus. So they're both like, they're sort of in the they're both in the executive suite, right? It's just like they're different, one's, CEO, CTO, whatever they are, right? But they're like in this high-level position, and they communicate with one another. And so behaviorally, damage to either the hippocampus or prefrontal cortex in spatial navigational tasks gives you very similar, deficits. That is, you need both in order to make spatial decisions, to navigate in space, use space to navigate. You also get episodic memory deficits. And so just using this kind of a simple T-maze task and then looking at recording simultaneously in both. And now back to this sort of, this figure. And in this case, when I was mentioning to you, these recordings are taken in the prefrontal cortex. These are in the hippocampus. But you notice cells in the prefrontal cortex are phase locked to the hippocampus. So at a minimum, you say, look, if this is a code, if some downstream is actually using it, they have, at a minimum, at least have to, they have to be able to synchronize to this, right? To the if there's a temporal, sort of coding dimension, you have to have that sort of temporal coding dimension available. And so phase locking is a minimum requirement for, the decoder, for a decoder downstream. And the prefrontal cortex seems to be expressing that. It's phase locking. Cells here phase locking the hippocampal theta oscillation. And if you kind of ask the question, well, what would the phase locking be? So you kind of phase lock when you want to communicate, when you would want to decode. It's like listening to a radio station. You would tune into a radio station when you actually wanted to hear the content. And so the idea that perhaps this relationship is one that is dynamic. Because while the hippocampus anatomically is like permanently connected to the prefrontal cortex, functionally, the prefrontal cortex, it's an executive. It's not like it's only listening to one, to the one CTO, probably very rarely doing that. Most of the time it's processing input coming up, from the bottom up. And so there's going to be some selective engagement, selective communication. What would that look like? And given that this sort of the synchronization would be a necessary condition, you can also think of using that as an opportunity to enforce like a sort of dynamic functional connectivity. And this is, one idea that a lot of people have advanced. And so the idea that you could use synchronization mechanisms, perhaps even oscillatory coherence, to impose this sort of dynamic functional connectivity. The hippocampus and prefrontal cortex only communicate when you have this phase locking. And so the question is phase locking, in fact, dynamically coordinated? And in this task, just the short answer, where this is a spatial working memory task. Animal starts at one of these two arms, it runs down this arm, and in this direction it has to make a choice. And we can make it like a simple match to sample. If you started on the upper arm, when you get to here, you got to go to the other upper arm, right? If you started here on the lower arm, you go to the lower arm. So the choice that you make here depends on where you started from. Then the animal turns around, it runs back, and in this direction, there's a little door here, and we just choose for it. We force the animal to go one way or the other. So the task is behaviorally symmetric. It's like running up and down this little corridor, but in this direction, the animal chooses. In this direction, we choose. And when you do this, what you find, and then you look at measures of like coordination, coherence, it's like the timing of prefrontal spikes with respect to the theta oscillation, for instance, here. Result is, and this was working with Matt Jones when he was a postdoc in the lab, that you get this theta phase locking in the prefrontal cortex, prefrontal cells locking to the hippocampal theta oscillation under very particular conditions. And that is when the animal's in the central arm, going in the choice direction, and makes the right choice. So effectively uses information about where it came from, and that's this bar here. So when the animal's going in the choice direction and it gets the choice right, this is reflected in significantly increased coherence. So in a sense, if I'm just looking at the brain, I'm recording these two areas, and I see, oh, look, theta oscillation, look, there's a lot of coherence right at the choice point. I can predict, sounds like the hippocampus is talking to the prefrontal cortex. I think he's going to make the right choice. And then bang, animal makes the right choice. On the other hand, animal gets the choice point. There's like theta oscillations in both, but they're not coordinated. It's like, it doesn't seem like they're communicating here. I don't think the animal's going to make the right choice. And indeed, so you could predict choice behavior based upon the degree of coherence between the hippocampal and prefrontal cortex. In the forced direction, where the animal doesn't make any choice, no coherence. So it's like, it's not, it could be, there certainly could be coherence, no reason why there shouldn't be or couldn't be. It's just there's no utility because the prefrontal cortex is not directing the behavior in this case, it's forced. So in this example, this is just like the one I gave is the match, you go to the same arm. You can do the same thing where you you try to reverse it, change the contingency. So like now, this is what's referred to as like a reversal condition, where now animal starts here, but instead of going to the same arm, you have to go to the opposite arm. That's a little more complicated, especially for rats, but the idea and result remains the same, and that is it's the coherence that it's correlated with and predicts performance. And so now in the hippocampus, doesn't make any difference. Same theta, the degree of theta phase locking is exactly the same. Choice, right, wrong, forced, doesn't make any difference. And so the idea of the hippocampal coding, it doesn't distinguish me. Am I using this to make a choice? Am I just using this to predict where I'm going to end up next? It's exactly the same. Choice does not influence the degree of the phase locking. And then again, arguably, we sort of talked about this, the place cells and the theta sequences, while the theta sequences might actually reflect the choice behavior. What it's really reflecting is if there was a systematic bias in the trajectory, if past location accurately predicts future location, as every time I started here, I go to the right, then you get remapping different place cells, and indeed the theta sequences would correlate them. But it's not, that's retrospective. It says the hippocampus in the code is reflecting what you did before and is not really predicting what you're going to do. It simply reflects how the future was correlated with and predicted by the past, rather than some kind of transient trial, contingent behavioral choice. And again, theta power, all the same. So it really is this coherence that's the choice. That's the choice thing. Coherence, choice. Theta, behavior. Yep. In this task, you do not know. We did another variant, similar to this. It was like a figure 8 maze where we could change the behavior so that now, when they're doing this, the past is always predictive of the future. I mean, if you're doing it correctly. But in a variant, this is like the reversal where you change the contingency. So now, the past no longer predicts the future. What you see is there's an interesting transition, transient in place cell, response. So what happens is the place cell representations, when animals become familiar with the task, they tend to be shifted in the prospective directions. The place cells tend to, kind of shift into more of the firing rate is in the future direction. If now you violate that, so now the future, you change the, correct behavior, so there's a reward component to it. What happens is the place cells will now shift more retrospectively, and that is the predictive component, the future component becomes weaker. they're more retrospective, and then as the animal picks up the right behavior, they become more prospective. And so it's like this, this retrospective, prospective, activity is not fixed. It's biased by choice, reward, and correct behavior. And so when it's more predictive, you get this more consistent prospective or predictive behavior. Now, how does that, what's the circuitry that accounts for that? How is this impacted by, as I mentioned, changes in the place cells themselves, as opposed to like the theta sequence-like mechanism? So very much like phase precession, there can be the two components. There's the individual cell component, the place fields, phase precession, place field, that's a single cell property. Then there's the theta sequencing, that's a population property. And so, how do those change when you kind of violate expectations? So now the future, your prediction of the future is no longer as valid. You see these very rapid changes in place field representations followed by changes in behavior. So that experiment was important because it showed that the changes in place cells and place fields is not contingent on behavior. The behavior was the same. The animal kept doing the same thing. It's just now they weren't getting the same rewards, and so they then changed their behavior. But as soon as the behavior was no longer being rewarded, in other words, the future was no longer meeting expectations, you saw this kind of remapping-like behavior change in place cells, which is sort of what we were talking about earlier. You remap when, stuff doesn't meet your expectations. Now, there's sort of an interesting corollary to that, which is something we're sort of interested in following up, and that is it suggests that things like remapping and changing configuration is going to be dependent on this prospective component. And that is, it's at that point in the this kind of late phase theta activity that may serve as the sort of the trigger for remapping, which is sort of an empirical question. But the reason that it's sort of an interesting, accessible question is that the follow-up to this experiment, which is just showing that you get the selective coherence when an animal is making a choice, and I should point out that this coherence, when it's expressed, it's like very specific. It's like right here in about the half second as an animal's running. It's right here in the half second just before the animal makes the choice. And then as soon as the animal makes the choice, coherence is gone. So it's dynamically coordinated, presumably, in order to inform choice. But once the animal has already made the choice, then prefrontal cortex doesn't need to listen to the hippocampus anymore. So it's something that is, the coherence is being regulated or modulated on a very short time scale, probably on the time scale of single theta cycles. And when I should mention, we sort of talked about like these theta sequences and how fast to transition. The Moses did a really clever experiment. This is the so-called theta flicker experiment. And that is, they had an animal in a box, and then they could project like visual cues so they could change the context like instantaneously. So it's like, oh, I'm looking here, I'm in the in a lecture hall, and bang, oh look, I'm out on the beach. And so they could like flip, and they could flip back and forth. And so what they found is when you flip, like go from lecture hall to beach, what happens is like on individual theta cycles, it'll be lecture hall, beach, lecture hall, beach. It'll flip back and forth. It'll flicker back and forth before finally settling on the context, the appropriate context. So there's like a period of uncertainty, which it sort of makes sense. It's like if I'm sitting here and all of a sudden everything's changed, it's like, wait, did I really get teleported somewhere? Or is this like, But what was interesting is that the alternation on alternate theta cycles, you could essentially flip context, single theta cycle. So the theta cycle was sufficient to actually communicate, presumably, right? Code and communicate contextual information. So the theta cycle is sort of the unit, the computational unit, which is used for, cognitive processing. Consistent with this idea here. Because, like 500 milliseconds, you basically get like 5 theta cycles. So the communication the hippocampus has with the prefrontal cortex is like you get 5 theta cycles to decide what you're going to do here. Like, where did I come from? Which way do I go? This is just to show, okay, you can say, look, you see this correlation, but is it really causal? This is an active manipulation. Josh Siegel went in and actively inhibited activity of these 2 phases. That's it. Just like late phase, early phase, What do you get? And same task. It's just now he's going to optogenetically drive inhibitory cell activity, either in the central arm or in this starting arm. And the reason for doing this is so he wanted to ask, like, is there a good phase of the theta cycle? What phase of the, you see this phase locking between in the prefrontal cortex and the hippocampus, but what's the actual period during which the hippocampus is communicating with the prefrontal cortex? And so the idea is, well, we'll just like block one phase and the other, and if you block the useful phase, the one that's the prefrontal cortex is listening to, you should see some behavioral deficit, right? It's like animal can't choose. So what he was looking for was some sort of loss of function, block the important phase that the prefrontal cortex is listening to, and you see a loss of function. Now, to do that, of course, you have to show that, well, like other phases, so you have to like alternate phase. So I do one late phase and early phase, and you have to show it's not just if I just block one phase, maybe just like disrupts hippocampal function. So let's show that it's actually specific to the choice period. So if we do the same thing here, early and late phase, two phases, two theta phases, two behavioral phases. Do the same thing here when the animal's just at the starting arm, right? It's not making a choice, it's just running here. So that was the expectation. You'll find the good phase. It's going to be like, you will find selective loss of, impairment of performance when you block the good phase only at this choice point. That was the expectation. And that's not what he got. That's not what he got at all. What he got was really kind of curious. He got an improvement in performance. So when he blocked the phase, activity in certain theta phases, he got an improvement in performance. But it wasn't a general improvement in performance. There was a double dissociation. There was an improvement in performance depending on the behavioral phase. And that is, in this case, when you block when you block early phase information here, when the animal's on the center, on what we refer to as the retrieval segment, you improve performance. When you block the late phase information on the encoding segment, you improve performance. So it's like, so that's kind of weird. Why are you improving performance, and why is the phase? So there's not a good phase. Which phase is good depends on which phase of the task you're actually on. And so in thinking about this, there's another really interesting kind of biophysical feature of the hippocampus that's important to note, and that is, so looking in area CA1, which is where we're recording, area CA1 gets input from that trisynaptic loop that I mentioned, and information comes from the entorhinal cortex, through the dentate gyrus to CA3, CA3 to CA1. So this is the classic trisynaptic loop. But there's also a direct pathway, directly from the entorhinal cortex to CA1. And so in CA1, you're actually getting like two sources, one from CA3, one from entorhinal cortex. And those two inputs, they're both theta modulated, but they're phase shifted by 180 degrees. So the hippocampus is getting two sources of input, but they're coming in at different phases, early phase, late phase. So it's now you start to think, oh, that's kind of interesting. There was like this phase, early phase, late phase thing with temporal coding, rate coding, past, future, and now you have like entorhinal CA3. Oh, maybe they're actually related. And so maybe what you have is like those different phases, functional phases, right? The temporal coding, predictive, is actually associated with a different set of inputs from CA3. So CA3 is, this is where I think you're going to end up. The early phase, the entorhinal cortex, direct inputs like, CA3. Like, I'm going to tell you, this is where you are, right? This is what you're getting. This is the present state. And then you can like rate code this with whatever confidence you think, okay? And so now you have encoding present state information, encoding future state information. Now you think about the task. Well, this task actually has sort of two components to it. For working memory, you have to first remember, where did I come from, right? So like, where am I, and then where did I come from? And then here, when you get to the central point, it's like, okay, given that, where do I have to go to? So there's like this initial encoding and then this predictive state estimation, two different components. This would be late phase dependent, presumably, it's the temporal coding part. This would be early phase dependence, like how confident am I that I started in the left arm versus the right arm? Encoding, retrieval, that's how I refer to it. But it's this idea there are two different functions, two different phases, and now you say, why did you get an enhancement? Why did you get an enhancement when you blocked the other phase? So you can say, look, the hippocampus, it doesn't know that it's doing any task here. Like you're starting here, it's doing encoding, and it's also doing retrieval. Say, oh, I'm here, and I am predicting that you're going to end up going down this arm. So they're like these sort of two components. Hippocampus is constantly broadcasting. By suppressing the irrelevant one, so here by suppressing the predictive component, the late phase information, it means now this early phase, the encoding component, is the only one left. It's dominant. So you're essentially biasing, you're biasing for the task relevant component, something that presumably the hippocampus is not normally doing. It's doing both because it doesn't know doesn't know anything about task. It just knows both of these pieces of information are likely to be useful at some time. And so the experimenter here is kind of tilting the scales based on the task that it wants the animal to perform. And that's why you get, and so in this phase, when you're doing retrieval, it's like it doesn't make any difference that, do you have an accurate code for the central arm? There's nowhere else you can be. This is it. So that kind of present state, the early phase information, is not really relevant to the task any longer. So that's the idea. Different phases carry different information driven by different sources of input. And so essentially by suppressing the entorhinal input, at this point, you bias for the CA3 component, giving you better predictive output. By biasing against the CA3 component, it gives you better present state estimation. And then just a little observation. So you think, oh, okay, so that says that like downstream, prefrontal cells can tune into these different channels. So maybe you could say it's like an encoding effect, or it's just like a downstream effect. Maybe downstream what you have are different prefrontal cells listening to different channels. Where am I now? Where am I going to go in the future? And so if that were the case, what you should have is prefrontal cells that have different preferred phases, and that's exactly what you get. Prefrontal cells actually have preferred phases across the whole 360. They're sampling all of the hippocampal phases. So some are tuned into the future channel, some are tuned into the present, past channel, and then depending on the task, you get this kind of bias. Oh, we're going to make a choice. Well, better, tune in those, prefrontal future channel, cells. So the idea of selective coherence, selective phase for task-dependent coupling. And okay, that's it. And now in the last 1 second, we'll do the sleep and memory. Okay, sleep and memory, yeah, they're a thing. Okay.